We begin part 2 of the endocrine system, page 594. We have this diagram right here, which indicates a target cell or tissue with cells, and we're noticing that on the cell we're having the hormone of interest here reach across space, the matrix between cells, the interstitial spaces, and bind to a receptor. So this is the initial receptor. Here is being represented an entire cascade of energetic processing until finally we get to what we call here a second messenger system. The purpose of this second messenger, in this case cyclic AMP, that we are derived from ATP. It's a triphosphate. We get the two phosphates removed to a single phosphate. The cell more or less holds its own hand and creates a little circle, so cyclic AMP. And the purpose is phosphorylation, so we activate a protein kinase. The protein kinase phosphorylates internal molecules within the cell, and those molecules enter the nucleus, dock on the DNA at a promoter site, and activate genes. So that's the purpose of this whole system here, is that we engage in a process in the body. Perhaps we've had a run in the morning and we've enjoyed our day so far with a little bit of distance running in a beautiful park, and we have some endorphins released and some serotonin, and that activates cells throughout the body and tissues throughout the body to wake up, improve their system, and so on. So second messengers in this case usually is the result of some bodily activity. Okay, so the in this case we also note here on the next page that a second messenger could also include a calcium molecule. The PIP2 calcium signal works similarly in the sense that it's going to activate phosphorylation and create a second sort of messenger system that again causes some phosphorylated molecule to enter the nucleus and cause further activities of the cell. That takes us then to this next mechanism. We saw mechanisms on the previous page wherein we arrive at a cell surface and we find a receptor that is extremely specific for that molecule of interest. So in this case we have something a little bit different. One could even say a little bit more powerful or invasive in certain ways, which is that all steroids, whether it's progesterone, testosterone, estrogen, or some of the others that we'll be looking at, like aldosterone and cortisol, any of those will actually pass right through the membrane unimpeded. There's one non-steroid that does that as well, and that's thyroxine. So the mechanism here is that any of these molecules will actually pass right through the membrane. If it happens to be on the external skin, we have skin patches for doing this very same thing. And in fact, the skin has its own steroid molecule that we need to think about called vitamin D. Vitamin D is a steroid molecule that gets activated at the kidney. So that's something that's of significance, consideration. And again, we can pass through the membrane here. What happens is that there is a receptor already present in the cytoplasm. The receptor and the hormone combined, we enter the nucleus without phosphorylation or the need of it, and we dock on the DNA at a promoter site and activate a gene sequence. Okay, so the result is the expression of some phenotypical outcome for this particular cell. Okay, so that would be the idea here. Then we press on here to this next idea. And we discuss interactions of hormones. There are hormone interactivities. For instance, estrogen is required for other things to take place. For instance, once there is an estrogen, how shall I say, priming of the human body and adequate amounts of titer, then that means that receptors will be made for, it might be hard to read that, oxytocin. Okay, so that just means that, for instance, a pregnant mom will have receptors for oxytocin produced on her uterus, as well as on the myoepithelial cells deep within the breast tissue for milk letdown. Okay, so just any adequate amount of estrogen. It could be also at mid-cycle, such things are experienced. And so there'll be a special experience at that time because extra receptors are being created and are present. Then we have something called synergism, and there are times when estrogen acts alone, and there are times in the second half of the cycle when estrogen and progesterone act together, and entirely different things developmentally will happen during the second half of the cycle after ovulation. So there's development of the endometrium in a completely different way that we'll take a look at when we get to that section of the book. And then we have, how shall I say, antagonism. Normally we think of antagonism very much between the parasympathetic and the sympathetic. Occasionally with the sexual cycle they work together, but oftentimes they're antagonistic. Here I'm offering you a different view. Insulin is going to be present by day, and it's part of the daytime cocktail. Glucagon generally is considered part of the nighttime cocktail and is present to produce extra blood glucose. So here by day we acquire food, we input the glucose and other nutrients into our body. We have storage facilities for the glucose as glycogen or fat. And then by night we have glucagon, which then takes from those stores and delivers to the growth hormone, which is doing a tremendous amount of work during the nighttime. Okay, then we move on to this next stage in which we consider this relationship now between the hypothalamus and what we commonly call the pituitary. The hypothalamus up here, the pituitary down here. Once you've learned the interior of the skull and you've visited with the sella turcica, which is just behind the optic chiasma, then you discover that the pituitary sits in the sella turcica. So that's what's going on there. We have this little bridge right here called an infundibulum. They're calling it an infundibulum. Notice right here we have the optic chiasma. You might recall that in the Turkish saddle, in the anterior aspect of it, there's a groove right in the front of the Turkish saddle, the sella turcica, a part of the sphenoid bone, and in that groove resides the optic chiasma. We learned that this is where the medial side of each retina crosses over to the other side of the brain for processing by that portion of the brain that brings us to the occipital lobe. Okay, so we'll recall that. We'll also recall that actually this is the moment that we would think to ourselves that there's an activation and release that occurs right here whenever there's light and light goes away, that we get melatonin. So we're going to be having a group of population of cells up here that communicate down here with this population in the posterior pituitary that's referred to as the neurohypophysis. Okay? And they actually have their axon terminals extend all the way down. So their cell bodies are up here, their axon terminals are here. and I'll pick that